Good evening, everyone. Welcome. Please come on in, find some seats, plenty of space. All right, thank you so much for being here tonight. I'm Beth Coakley. I'm the Dean of Mills College at Northeastern University, and I'm thrilled to welcome you to the Chan-Norris Scholars Distinguished Lecture featuring Dr. Nick Pilfold. This lecture series is a cornerstone of the Chan-Norris Experiential Learning Program in Science and Sustainability. This Mills program creates exciting learning opportunities for students here on our Oakland campus. These experiences are grounded in science, sustainability, and conservation. In this, our inaugural year, our program theme is Emerging Infectious Diseases, and we have an incredible cohort of Chan-Norris scholars joining us this evening. Already, these students have visited the Marine Mammal Center, gone behind the scenes at the Cal Academy of Sciences, and met incredible scientists. We're very grateful to Annette Chan-Norris, class of '65, and Evan Norris for making this program possible through their generous donation. And now I'd like to introduce the heart and soul of this incredible program, our inaugural director, Dr. Professor Swope. Dr. Swope has been with Mills since 2011. She's an evolutionary ecologist and studies 3 very rare plants in Marin County. She's interested in how plants cope with life in a variable world and the role of genetic diversity in promoting local adaptation. Dr. Swope uses the data she collects to make predictions about whether these species are likely to evolve fast enough to keep up with climate change and also to recommend ways to protect them. She's also developed really cool courses on systematic botany and fire ecology, which is especially poignant for us in California. She's even taken groups of students to Ecuador to study conservation in the Amazon rainforest. From her research to her teaching to her directorship of this program, all of Dr. Swope's work has a strong conservation theme running through it. Please join me in welcoming Dr. Sarah Swope.
Hi, everyone. Thank you very much for being here. So this is the second of our distinguished lecturers of the Chan-Norris program. And as Beth mentioned, the theme for the Chan-Norris this year is Emerging Infectious Diseases. So for that reason, I'm very excited to have Dr. Nick Pilfold here to talk about his work on, he's going to talk specifically about his work on polar bears, climate change, and emerging infectious diseases. So Dr. Pilfold got his bachelor's at the University of British Columbia and got his PhD in ecology at the University of Alberta, right? He studies large carnivore conservation. I have to get the name right. He's as a scientist at the Conservation Science Wildlife Health at the San Diego Zoo Wildlife Alliance. And he's going to talk a little bit about his work and what all that means. But ultimately, his work is at the intersection of basic science and effective conservation, which is sort of where the rubber meets the road and what I'm particularly interested in, which is why I wanted him to come talk to us. I think his work is going to be really interesting to all of you, no matter what your interest is. He's going to be talking about serological analyses that he uses to detect pathogens in polar bears. So if you envision yourself going on to do, say, medical research, there's something in this for you. This, of course, ties to demographic data and population declines in polar bears. So if you're an ecologist like me, there's something in it for you. And then, of course, all of this is obviously of great importance from a conservation standpoint. Okay, so Dr. Pilfold is going to talk for about 45 minutes, and then there'll be a chance for all of you to ask questions here in front of everybody. And then afterwards, there will be a reception in the lobby where you can talk to him in a little bit more informal setting. Okay, and with that, I'm going to hand it over to Dr. Pilfold.
So much. Thank you. Am I coming through okay for everybody? Good. Well, thank you so much. I'm going to go back to the top here. I know it's day after Super Bowl, so I appreciate you all being here. Harbingers of a warming world, not necessarily the most cheery of chats, but I promise there'll be a lot to take home from this. And what I hope for all of you in here is that there are 3 things that you may remember from this lecture. The first is actually that zoos can be leaders in the conservation of wildlife. Zoos have had a very long history, even going back to ancient Egypt. That was some of the first zoos. They've often appeared alongside empires. The Romans liked to have zoos. Apparently there was a polar bear that was kept in the London Tower at one time. And zoos have had this history that has been mirrored alongside museums. They're collections to show everybody what we brought from the rest of the world in so you can see. They mirrored the language. So if you go to a museum, you might meet a curator, say a curator of ancient antiquities who's brought relics from ancient Greece. If you went to a zoo, you might meet a curator of mammals or birds who's brought the birds from the world for you to see. And in the 20th century, there was a lot of work that was done at zoos to make the facilities better for the animals that live there. It made the habitats more realistic to where those animals had come from. And now zoos around the world are leaders in longevity for species that live in their care. But in the 21st century, we have to continue to move forward to make this institution relevant, and that's through conservation. And what I hope to show you tonight is how you compare ex situ and in situ care. Ex situ is Latin for out of place. That means the animals that we have in our care at a zoo. In situ is the animals that live in place or in their habitats. The second thing I want you to take home is a consideration that a career in conservation is a very worthwhile endeavor. You are among the best and brightest of your generation, and you have careers ahead of you. I hope that you'll consider, after watching my talk, potentially a career that influences conservation. Right now, we're very wrapped up in the human element of our lives and the global political chaos in some ways that's occurred over the last few years. So if you open up your news, we're always talking about human-related elements, things that strife that we're trying to get over ourselves. But we need people who will give a voice to the rest of the species that we live with on our planet, because without us, they don't have a voice. And extinction is the end of a very long process that we do not wish upon any species. And the last thing I hope you take home is that the natural world is rapidly changing. So whether or not you take those first 2 points to be true at the end of my lecture, no matter what you end up doing, the world that we know now is not the world you're going to know when you're my age. And it's not going to be the world you know when you're in your rocking chair and the robot is serving you tea. It's changing very, very quickly. And I think probably much of your course material will show you that. But no matter what you end up doing with your life, you have to be prepared for the fact that the natural world is changing and we need to adapt. Okay, so let's talk a little bit about where I'm from. I'm from the San Diego Zoo Wildlife Alliance. What that is a conservation NGO with 2 front doors, the San Diego Zoo and the San Diego Zoo Safari Park. Just by a show of hands, has anybody been to San Diego Zoo? Oh, good. Okay, so many of you know what I'm talking about. Between those 2 institutions, we welcome about 5 million visitors every year, and it gives us the opportunity to make connections between people and wildlife that they would otherwise never meet, and hopefully an emotional bond for them to care, and an understanding of what the conservation threats are. We apply our expertise in animal care, veterinary science, conservation science to 8 global hubs around the world where we're trying to protect species in situ, in place. So that includes places like East Africa, where we have a rangelands program, includes the Amazonia program in Peru, a Southwest program right here in California, and the Oceans program, where we conserve polar bears, where our work is there. And if you've been following the slide and counting closely, I said there are 8 hubs, but there are 9 images on this map. And the idea is for 7 of these hubs, they are geographically focused, but for the Oceans hub, the link between all the species there is that they depend on the marine environment for their survival, and that's certainly true of polar bears. Okay, let's start talking about polar bears. This is their home, the sea ice of the Arctic. When it's at its maximum extent, which is coming soon, this is last year's image, but in March we'll see the maximum extent of ice again for the year. It's approximately 14.3 million square kilometers. Absolutely huge area. We estimate there's about 23 to 26,000 polar bears that live in this area. And if you look at the way the bears move and behave, they spend about 95% of their life living on that sea ice and depending on the marine system below it for their survival. But this is a system that's undergoing immense change due to climate change. I'm sure you're pretty well aware of what's happening to the Arctic sea ice. As we put out more and more greenhouse gases, the atmosphere continues to maintain the heat in the planet, not letting it dissipate out, and that's melting the sea ice. So what you're seeing right now is a satellite images of the minimum, which occurs in September, and the loss in the satellite era of that sea ice. When it used to be around 6.5, 7 million square kilometers, it's now down almost half of that in that time, just in the interval over the past 40 years. So polar bears call that home. It's really a simple equation for a bear, right? It's their primary habitat, and they're losing that habitat as the planet warms. For polar bears, it's really an energetics argument for why a loss of sea ice is a threat. So if we think about the annual cycle of the bear, right now, out in the winter, they're hunting seals. This gets really good for them in the spring. And then come summer, when that ice melts out, in most places in the Arctic, the bears are forced onto land. Well, they'll spend their time until the ice reforms, and they go back out. So what climate change is doing is it's pushing bears on two ends. It's making this period that they're on land longer, which cuts down the amount of time that they're able to be out on the sea ice hunting seals, and increases the amount of time that they're in a fasting state. So when they're on land, they're not consuming calories, they're just relying on the reserves. And those reserves are built during the ice season. So less time to build up the reserves, more push for those reserves because you're spending a longer time on land. It costs bears about 1 kilogram a day to be on land. So it's good that they're a big animal, because can you imagine losing 2.2 pounds every single day just because you weren't eating? It's a lot. And it affects the younger bears more than the bigger bears. So this is some work I did a few years ago where we had groups of bears. These blue lines here are the adult males, and the gray and black lines are subadults. And we looked at the size of the bears and looked at their mass change while they were fasting. And then we calculated if they were continue to fast, how long until they would starve. Just a model. So here's 100 days. There's 200 days. When you get out to 200 days, you can see that the male, big males, the adult bears here, 15 to 20% of them might not make it out to 200 days without food. But if you're a subadult and you're still growing, you're a younger bear, cumulative proportion of those bears that wouldn't make it, 70, 80% of the bears. This is one of the mechanisms by which climate change operates for polar bears. Adult survival of polar bears is actually pretty good, but the younger age classes are affected more. If you're a subadult and you're trying to be recruited into the adult population, you face increased energetic trade-offs because you're growing. And for adult females, they have mechanisms of survival built in for reproduction. So when they don't have the right body condition, they just won't reproduce. And in some ways, this is what we call a silent decline. It's just less bears. Okay, so where can we at San Diego Zoo help with these questions that we have for their wild counterparts? You might be looking at this image going, what in goodness name have you done for this bear? This is Takik. In this image, she was 16 years old. Takik right there is sitting on a treadmill. That treadmill is encased in a plexiglass enclosure. Why would we do that? Well, we want to measure oxygen exchange inside that treadmill enclosure when she's walking. This took a lot of work from our wildlife care team and a lot of peanut butter. So everything is done with reward, and Takik would get into this because there was peanut butter at the one end and walk on the treadmill because there was a spoonful of peanut butter for her to go to. And when she didn't want to walk anymore, she would just sit her butt on the treadmill and like ride it to the back and then just start over. So it took about 6 months for Takik to get used to the treadmill and for us to have good data. Now, why do we care about how much energy a polar bear burns while it's walking on a treadmill? This image, the bottom right of your screen, is a tremendously wide swath field from the North Slope of Alaska, the Beaufort Sea. This is Canada, that's Alaska. So this is hundreds of thousands of square kilometers. What you're seeing here are daily images that are stitched during the breakup season. And what you'll start to notice is that ice fractures very, very quickly over huge expanses, hundreds, if not thousands of kilometers long, these leads will form. Now, what we know about bears from their movement patterns is that they walk against the flow of sea ice. They want to maintain their place in space and time. They don't want to drift out into some place where they don't want to be, so they'll walk against that drift. What's happening with climate change is the ice is becoming thinner, more fragile, and more mobile. And so essentially what we're doing is we're taking that treadmill and we're turning the speed up. So Takik tells us exactly what the energetic cost of that is for her wild cousins. Another amazing thing that we couldn't do in the wild. This bear here is listening. Oops, sorry, pardon me, wrong button. This bear here, pardon me, restart it. Okay, there we go. This bear here is listening to a tone at a certain frequency. When she hears the tone, she'll touch this pad, and she'll get a reward for it. So she's telling us at what frequency and range can she hear, and it's creating what we call an audiogram. Now, what we do with that is we can understand what the disturbance is for bears who are in dens near industrial areas. So on the North Slope of Alaska, there's a lot of oil and gas activity that occurs near polar bear maternal dens. So we went to those dens, we put microphones inside the dens, and then we measured the noise at different distances from different types of machinery. And this is just one of them. This is a helicopter passing over. That's the noise it makes at different distances relative to inside the den. That red line is the polar bear hearing threshold at that frequency. So we know that once that helicopter is within about 600 meters of that den, even in a closed den where the snow is already still built around the front, that bear can hear it and possibly be disturbed by it. This kind of research is the research that U.S. Fish and Wildlife relies on to keep oil and gas at a certain buffer distance, saying you can't come in any closer because we know that the sound from these machines is going to disturb bears in their den. The other thing we can't do very well in the wild is know exactly what's happening inside the den. I'm not going to go and poke my head in and say, hey, what's going on? But in a zoo, you get unparalleled access for when cubs are born. And so we've been taking video data of these first few years of birth, few days of birth when they come out. And when the cubs come out, they're like the size of a loaf of bread, and they're about a pound, and they're blind, and they're deaf. They're completely what we call altricial. They're dependent very much on their mothers. But from those particular videos that we have of the mums raising their cubs, we can understand the role of communication, or what frequencies are the cubs communicating with their mothers, the energetics of the care, and that gives us a window into a place that's very critical for a polar bear, but we'd never be able to get to in the wild. Okay. Moving to the wild, along the idea of doing maternal den surveys, we've been working in a place called Svalbard in Norway. It's a high archipelago of islands. And we've been placing cameras outside of dens at a safe distance so we don't disturb them, and watching what happens when the bears emerge out of these dens. So this is one of the best denning areas in the world, and they den right into the snow slope, and then they break that snow slope out, and they walk out with their cubs, and they spend time outside of the den before they migrate. So what we're trying to understand is, what are they doing outside the den? Doesn't matter what the environmental conditions are. Have they been pushed out of that den early because they've been disturbed? Following along the theme of cameras, this is me and my PhD student, Holly Gamblin, and we're placing remote cameras out onto the sea ice. Now, why would you want to go out in this kind of weather and place remote cameras out onto the sea ice? This is about minus 25, minus 40 with the wind. And I'll tell you a little bit what she's doing right there. But we have these cameras placed because of this. So I told you polar bears depend on the marine system. What they're depending on is seals. That's their main prey. When polar bears eat a seal, they don't eat the whole thing. You can imagine it like, the way I like to imagine it is like a banana. So when they kill a seal, they peel the banana, they open it up, and they eat all the white stuff. That's blubber. So they eat all that fat, and then they leave the rest of it behind. And unlike other bear species, they don't cache their kills to come back to later. They just leave it on the ice. And so what we wanted to understand was, what are all the other species that might benefit from polar bears doing this activity? Because there's no other animal or process in the Arctic that can duplicate what a bear does, which is to pull a seal from the water column up onto the ice, rip it open, and then leave it there. And so the first thing we did was we tried to estimate how much is left behind. That's that paper that was just published. Our conservative estimate is 7 million kilograms of carrion every single year polar bears are leaving behind. The cameras are to figure out who's there, right? Who's coming and benefiting from it. We have an idea, but we want to know for sure. A lot of Arctic fox. We got our avian community, snowy owls, ravens, mums with cubs. It's actually very important resources for certain demographics of bears. Mums are energetically challenged at this time of year, so if they can find a leftover bit of seal, they'll take it. And so this is a mum that's just come out of den. You can tell because she's got big, oops, sorry, hang on, jumping ahead, pressing the wrong button. Let's try this again by going back. I'm sorry, my apologies, because the button is counterintuitive to me. Try this one more time, all the way back. Okay, don't want to ruin the moment. Right. Big chunks of snow on the back of the butt. That means she's just come out of den, okay? And so she's just out of come into den. Who's going to be with her? That one. That little guy's about 3 months old and very curious. So he'll make an appearance here in a second. Ah, there he is. And then there's videos of whatever this is. This is a bear, I think, drying itself. So we get all sorts of good stuff outside of the core science that we're trying to get. So in addition to those kind of questions, we also do ongoing monitoring. And this is where we're going to get into the serology and the pathogens. So to do ongoing monitoring, we have to track polar bears from the air in a helicopter. There's the bear there. So we're following footprints in the snow as we track that bear to its location. And then once we've chemically immobilized the bear from the air, we start to do morphometric workup and take biological samples. This is another PI that I work with, Dr. Andrew DeRoche from the University of Alberta. And once we've finished the workup on the bear, we're always making sure that that bear is in a healthy state before we leave it. So we've gone through, we've collected blood, some hair, some skin samples, a bit of fat. And what I'm doing here right now is I'm taking the read on the breath rate for the polar bear to make sure that it's still at the right level of sedation before we leave it. You might have noticed there was a big number 8 on the back of that bear. That's not for giggles or fun. It's a washable crayon that stays on for 2 or 3 weeks that we put on the back of the bear. And that's because you don't know the bears from the air individually. So if you fly over that bear and you see number 8, you're like, oh, we've already done your checkup. You're done. We don't have to see you again. One of the things that we're doing is putting transmitters out, ear tag transmitters that provide a GPS location once per day. And what you're seeing here is this is the coastline of Hudson Bay. This is the sea ice area, the bay area. This is the land area. And so when you see that spray of color, that's when we put the transmitters out. And then the bears are walking on that ice until it disappears. And once it disappears in the spring and summer, they start, boom, come straight onto land. So transmitters are able to tell us where are they coming back onto land, what's the phenology of what they're doing. And importantly, can give us real-world conservation results like creating marine protected areas. So you may have covered this at some point in your class, but we're about halfway through the decade for the 30 and 30 challenge. The nations that signed up for this at COP15 said that they were going to try to protect 30% of their land and 30% of their water by 2030. We're kind of working our way there. The Arctic is underrepresented. And so what we did with the tag data is we created a marine protected area boundary for suggestion of where that could be along the coastline of the Hudson Bay. We also suggest that in this case we should be using a dynamic marine protected area. That's one that shifts over time and season to best protect the bears. The bears in this case are an umbrella species for the rest of the sea ice ecosystem. So that's one way that all that tag data can be used for conservation. Okay, so that is just sort of a real brief kind of background primer about kind of where I come from, what I do, why that works, how you can take ex situ and in situ conservation data and combine it for the best practices for polar bears and their conservation. And now I want to dive a little bit more in detail around pathogens and climate change in the Arctic, and this is something that we worked on as a project over the last few years. So why would we want to do this in the Arctic? Why is it very important that we monitor diseases in the Arctic? The Arctic is warming at 3 to 4 times the rate of the rest of the globe. Environment is changing rapidly, and that could bring about disease outbreaks that threaten both wildlife and the people that live there. There's a number of mechanisms that this could take. One is called a thawing freezer. This is where you have melting permafrost, which can release pathogens that have been held stasis in the soil, sometimes for decades, sometimes for centuries. Anthrax is one that can be held in stasis for centuries. It's quite scary. And it can create thermokarst lakes. So what a thermokarst lake is when the permafrost melts, you get depressions in the soil, and all that fresh water accumulates into those areas, and they can become new point sources for waterborne pathogens. Wetter and warmer is a trend in the Arctic, and that's a trend that we expect around the globe will facilitate the survival and transmission of certain pathogens, and it's certainly occurring in the Arctic, so that's another mechanism. And then lastly, you have range expansion of temperate species. Temperate species are moving northward as the conditions change in the Arctic, and that can create novel interactions where they're bringing diseases that Arctic wildlife don't have immunity to or have never been exposed to, and that can create nasty outbreaks. So human health in the Arctic is also something we need to pay attention to. It is a unique place. There are key exposure risk factors. One, the biggest one, is the dietary consumption of wildlife. So many local people who live in the Arctic, they're subsistence hunters. So they utilize the wildlife for their own food source, much higher than anywhere else. And so that creates an exposure risk. Water supplies can be tricky in the Arctic. So if you think about the permafrost issue that I talked about with melting permafrost, if you were to build a settlement or a town on something that feels very stable and then that melts and shifts, then things like sanitation and water are difficult to maintain because the entire ground is shifting below you. Biting insects are a real deal in the Arctic for anybody who's been in that region. So much so that caribou have an evolutionary strategy to avoid biting insects. You'll see them in the summer get as close to the coastline as they possibly can. And that's not because they're annoyed that they're being bitten. That's because the insects carry disease. There's actually an evolutionary response to their migration patterns that's around avoiding insects. And if you get the right conditions in the Arctic, you can get real plumes of insects that crop up. The impact if you do have an outbreak, food insecurity and financial loss. So if you're a subsistence harvester and you harvest muskoxen and a whole bunch of muskoxen die, then that's literally your food. And if a bunch of animals have just dropped dead, you're not necessarily going to want to hunt the one that's still standing, right? Financial loss, if you have herds of reindeer or you run trap lines, and that contributes to your finances, then you can lose that. More importantly, I think, is there's a disruption of cultural practices. So many people who live in the Arctic are very attached to the land, and their cultural practices are about going out onto the land to continue their way of life. And sociologists have termed the effect of what disease can do in that landscape as ecological grief, because people are losing their wildlife and losing their way of life at the same time. Then there's the cumulative and cascading effects on the system. So we can never think about these outbreaks as a single point in time and space. If you lose a bunch of muskoxen, then you also lose what those muskoxen were doing, which is they're grazing, they're herbivory. And that's actually been shown in places where muskoxen have died. It's actually been shown that the types of shrubs that come back after they disappear are more likely to cause the type of environments that increase the rate of permafrost melt. So there are these cascading effects that happen, kind of like dropping a rock into a pond and having the ripples go out. So it's really important that we think about this in what's termed a One Health concept, which I'm pretty sure you've likely been introduced to, and that's where we take environmental health, human health, and animal health, and we think about it together. How do these things interact? Serology is a fantastic monitoring tool for us to try to understand how things are changing in the Arctic. So we'll review that really quickly. Serology is a measure of exposure to pathogens through the detection of antibodies that are circulating through the blood. And so what you're seeing here is a tube of blood that's been spun, and the serum has been separated out. And you can think of the serum as sort of the distillate. It has all of the antibodies flowing in a concentrated form, and so we use that to get a good read of the type of antibodies that are in your blood system. Antibody signatures have a variable length of presence in the blood, and we all know this from being vaccinated. Some illnesses that you're vaccinated against, you have to be vaccinated repeatedly in order to keep your antibodies high. Some, you can be vaccinated once and have antibodies for the rest of your life. So you have to take that into consideration when you're using serology, is that if you're looking at a time signature going into the past, there might be different lags in which that exposure had occurred. It might not all be the same time. This is really important. Serology is not a measure of the virulence of the pathogen. It tells us nothing about whether a disease developed in the animal or an infection or whether the animal died. It just tells us that the system had an antibody response. It was exposed to a pathogen, the antibodies are there. Okay. Why are polar bears really good sentinel species for the Arctic? Well, first is that they're a wide-ranging apex predator, and they inhabit both the marine and terrestrial system. So they are exposed to potential pathways in both systems. They are subsistence harvested by local indigenous populations, so they are consumed in some areas of the Arctic. We have long-term biological sample collections that allow for really rare longitudinal analyses. So if you look at a lot of the serological investigations that have been done globally, not just in the Arctic alone, a lot of them are snapshots in time. So they went and collected a bunch of samples, they looked at the antibody response, and then they publish it. But they can't really tell you what's changing over time. And that's where polar bears can provide us with some insight. There's a caveat here about polar bears, is that they're ideal sentinel species for many reasons, but what is uncommon for them is to understand the direct cause of mortality. If you think about it right now, I like thinking about what polar bears are doing right now. We're just coming out of the high Arctic winter. Light in some places of the Arctic is just cracking again for the first time, just for maybe a few minutes over the horizon. Some polar bears in the world have been living the last 6 weeks of their lives in complete darkness. We're not, as researchers, going to go running around looking for every dead polar bear and do a necropsy to see why did that polar bear die. We have very limited information for understanding how polar bears come to the end of their lives. It's actually why we also have a great amount of debate about when polar bears split off from grizzly bears evolutionarily. Because when they die usually on the sea ice, and their bodies drift to the bottom of the ocean floor. So we don't have a good fossil record. So we don't really have that direct cause to say, yes, that bear died because it was infected with trichinella, and that is why it died young. Okay, so let's talk a little bit about the study design and some of the pathogens we looked at. So for this particular study, we went to the Western Hudson Bay in Canada because it is the longest studied polar bear population anywhere in the world. So it has that longitudinal sense. We looked at 3 sets of time, mid to late 1980s, mid to late 1990s, and the mid 2010s. And we had 425 serum samples that were roughly balanced between adult males and females, and as much as we can within over years. And we looked at pathogens that were bacteria, viruses, and parasites to try to get a bit of a spread. And what we did is we modeled any time we had a seropositive, or basically an immune response that we could measure in the serum. We measured those occurrences so that, yes, that bear has been exposed to both biological and climatic factors. And that's what I'm going to talk to you a little bit about tonight. So, just a quick refresh on the Hudson Bay itself as a part of the Arctic. It is a seasonal ice system, so the ice forms every November. It stays in the system until the summer, melts out generally around July. And when it melts out, the bears are forced to go onto land before then they go back out onto the sea ice. The system is under a lot of change, and I'll just walk you through this really quick. So this is the Hudson Bay. This dotted line here is the home range of polar bears. That's about 250,000 square kilometers per bear. It's a huge area. This is where they summer, in the hatched area here. The blue, green, and gray are the time frames in which we collected our samples. This is what the sea ice looked like in July in the mid-80s, mid-90s, and mid-2010s. So sea ice in July, a lot more of it was present in the 80s, and less in the 90s, and even less in the 2010s. But it's not just the loss of sea ice. We've also had changes in temperature. So a steady increase in temperature over time, in the summer in particular. And the amount of precipitation that falls during the summer months has also been changing. So that's been steadily increasing over time. And again, these hash marks are just where we were collecting our samples from the bears. So let's talk about a few of these to give an idea of the type of mechanisms that are at play. So let's talk about toxoplasma. It's a zoonotic parasite that is thought to be facilitated by wetter conditions. When we looked at it in polar bears, we found that they had the highest exposure in Hudson Bay than anywhere else in their range. And we also saw that each of these time series, they were becoming, there's more and more exposure amongst the bears. More of the bears were showing up with antibodies. And so we went from 44% of the bears in the mid-1980s to nearly 70% of the bears by the mid-2010s being exposed. We found that it was positively correlated to wetter summers. So this idea that's out there that it's facilitated by wetter conditions showed up for us. And we have inferred, that an intermediate host species are likely at play. So let me talk about that a little bit. So potential exposure pathways. You have to differentiate in disease between what is called a definitive host and an intermediate host. So toxoplasma, it's a parasite, and a definitive host is an organism that allows it to complete its life cycle, and in this case, shed the eggs back into the environment. And that's cat species. So that could be your domestic cats or your wild cats. In our area, it's Canadian lynx. An intermediate host is an organism that will have the cysts, the tissue cysts in them, but they will not complete the life stage and they will not shed it back into the environment. So to be exposed this way, you have to consume that individual in order to have exposure. So we looked at these potential pathways to see if they fit with what we're seeing. The first one was domestic cats in town. So there are towns, and we wanted to know if polar bears who spent more time in towns were more likely to be exposed to toxoplasma. Nope, no correlation there. So no direct exposure to the little kitties. The Canadian lynx haven't been in the area where bears come onto land for a couple of decades. We talked to a number of hunters and trappers in the area. They haven't seen many of them at all. So they're out. So definitive hosts don't seem to be the particular issue. Down here, intermediate hosts, this is a migratory one. It's a lesser snow geese. Comes to Hudson Bay in the thousands upon thousands in the summer months when polar bears are also on land. Potentially, if a bear were to eat a dead waterfowl that may have died from toxoplasma, then they would be exposed. However, I told you that 70% of bears were exposed. So this one is a partial. Potentially exposed to some bears, but we wouldn't expect that 70% of bears would get their exposure just from eating waterfowl. They don't eat that much waterfowl. So the last one here is a seal. And what's interesting is that disease experts have suggested in the Arctic that toxoplasma may increase in marine systems through surface runoff. And so what that might look like, and the reason that summertime precipitation is so important, is you have your rain here that's hitting during the summertime period, flushing out increased amounts of oocytes into the water, and then these are picked up in filter-feeding fish, invertebrates, which are then eaten by seals. And the reason that it's so important that summertime is the signature is that that's when seals are hyperphagic. They're feeding at their greatest weight or amount and putting on a lot of weight at that period of time. And then when the ice reforms, the bears come back, eat the seals. Some of those seals will have or have been infected by toxoplasma. Now let's try a different parasite. This is Trichinella. Trichinella is another zoonotic parasite that's capable of transferring between animals and people. Maintained in the Arctic through a unique cannibal scavenging cycle. So what that means is that Trichinella stays within the host tissue. It doesn't get emitted out into the environment. It wants to live inside the tissue. And what's interesting about Trichinella is it has a really low prevalence in seals, which is polar bear's main prey. So how is it that polar bears are being exposed? Well, we see a similar dynamic, much more than the mid-1980s. We have this jump in both the mid-90s and mid-2010s that it is significantly higher than what was happening in the mid-80s. We found that older male bears have increased likelihood of having a signature, and that feeds back into this cannibalism part. We'd expect that if cannibalism was maintaining Trichinella within the species, that male bears who are more likely to cannibalize others or scavenge remains of individuals that have died because of Trichinella, that would show up in the demographics. We found that it was positively correlated with warmer summers and winters. And this one was interesting for us because the Trichinella in the system is cold tolerant. So why is warmer temperatures helping a cold tolerant species? So winter cold. Ideal survival for a cold tolerant Trichinella parasite is between 0 and minus 20 degrees Celsius. The minimum temperatures in our study were between minus 25 and minus 30. And so you're imagining right now that something has died, has Trichinella in its tissue, and the only way that Trichinella is going to move to another host is for something to eat that, right? And it has a limited run. If it's between 0 and minus 20, that run can be quite long. But if it gets colder and colder, the Trichinella is not going to survive. Even cold-tolerant Trichinella doesn't do very well at minus 25 or minus 30. So in warmer, extreme cold, you actually get an improvement in the survival of Trichinella. And importantly, access to the carcass. So if you think about a carcass that's frozen down onto the ice that has Trichinella in it, to get that Trichinella into another individual, something has to come and eat that. Extreme cold locks those carcasses away from a lot of different scavengers. They're very hard to bite and masticate and get into. And in the case of seals, I've lost my point here. Very hard to get into. That's all I'm going to say. They're frozen to the ice, and they're tough to get into, and that's one of the reasons that the Trichinella might not move, because it's not being consumed at very cold temperatures. Okay, summer heat. This is also what we found, is warmer temperatures in the summer were likely to increase the prevalence of Trichinella. Warmer summers increase the likelihood of heat stress in polar species. Polar species have no problem with really cold temperatures. They do have a problem with heat. That can alter the endocrine system, which depresses the inflammatory response, and compromises immunity to parasite invasion. So that can have a twofold effect in the summertime. If you are already fighting an infection, you may have increased mortalities of individuals that are close, because they're close, then they're heat stressed, and that's it. So then you have more point sources in the environment of carcasses that have died because of Trichinella. The other way that this might show up is if you're a bear, what trichinella is trying to do is you've consumed something as a parasite is trying to get from that digestive system out into the muscle tissue. If you go through a period of heat stress, even if it's just a small period in time, it might facilitate that movement for the parasite. And then if the heat comes back down and your heat stress is over, the signature might be higher because you're now dealing with a parasite that's in your tissue rather than in your digestive tract. Okay, a couple more for you, and we'll wrap it up. Tularemia, this one's really important because a lot of people are infected in the Arctic, a lot of local people with tularemia. It's a zoonotic waterborne bacteria. It's transferred through biting insects from rodents and lagomorphs. And it can survive in frozen water and soil for months at a time. What we found in the exposure for polar bears is, again, this jump from the mid-1980s to the mid-1990s and 2010s. Polar bear exposure was positively correlated to the amount of time they spent on land. So this is a sea ice signature. Less sea ice, more time that they're on land, more time they're interacting with biting insects. And they were much less likely to be exposed if they spent significant time near the coast while on land. And I told you about what caribou do, right? When caribou are on land, they try to get to the coast because that allows them to avoid biting insects. So we saw the same signature in polar bears. Those who spend a lot of time near the coast were a lot less likely to be exposed to tularemia. So what's happening over time in the Hudson Bay? It's a very buggy place in the subarctic in the summer. But the emergence, there's a cycle. So in June and July, you get a lot of mosquitoes, and then those give way to black flies. And what's happened for polar bears is in the 80s, they used to come on land in August. So they were primarily being exposed to black flies and deer flies. They've started coming on land in July. They've been pushed earlier because the ice is melting earlier in the year, pushing them onto land earlier, and that means they're being exposed to mosquitoes. So a potential mechanism. So we wanted to see if that held up by looking at a different potential pathogen. So we looked at the California serogroup virus. California serogroup is a set of zoonotics, mosquito-borne viruses. They're transmitted through mosquitoes and rodents and lagomorphs. Sounds very familiar, right? It's the only difference between tularemia and the California serogroup is one's waterborne, the other one's mosquito-borne. The key for this one, the reason that we like to use it for Arctic change, is that there's a thermal limit to mosquito vectors. And so you can measure how much this disease is moving in the Arctic as a reference to how mosquito populations are also changing. Are we getting some warming that's bringing this in? So what we found is a bump, but because it's a single vector with mosquitoes, not as big as you saw in the tularemia. Again, very similar signatures. Polar bears were more likely to be exposed when the temperature was warmer in the summer months. And that is because water is not a limiting factor in this system for mosquitoes to breed into, just temperature. Polar bears were much less likely to be exposed if they had spent significant time on the coast, near the coast. So very similar signatures between the two, reinforcing the story around insect vectors. Okay, a few takeaways for you, and I really appreciate your attention through the talk. What we're finding here is that ecosystem level changes in the Arctic that are reflected in polar bears are more than just sea ice. So I'm sure you're all very familiar with the story and the story I gave you tonight, which is that as the climate warms and the sea ice reduces, that's the primary habitat for polar bears and the principal threat. And that's been the main focus for polar bear researchers too. We spent a lot of time looking at that interaction between sea ice and bears. But what I'm showing you tonight is warmer temperatures and wetter conditions also have an impact. The largest increases in pathogen exposure occurred again and again between the mid-1980s and the mid-1990s. And this was a period for the Western Hudson Bay polar bear population where they declined by a third in abundance. We had declines in reproduction and body condition. And then actually after the mid to late 1990s, things have stabilized out a little bit more. And you see that reflected in the way the pathogens are moving as well. And what this is telling you is, I told you before, we can't relate the severity or the virulence of any of these pathogens because serology doesn't do that. But what it is telling you is that the system's gone through a step change, and all the indicators are lining up in the same way. And then lastly, of the pathogens we've monitored, 9 in total, zoonotic pathogens, and all the ones I showed you tonight, were most likely to show increasing patterns over time. So again, you keep seeing the zoonotic ones, and that's the ones that can pass between wildlife and people, be the ones that are more likely to be increasing in exposure in polar bears as a sentinel species. And so we need to be very aware of that. I want to touch on this one as my final slide, which is when we published this paper where most of this information came from. It was 2021. And we all think back to 2021. We were in the middle of COVID-19. The world was on edge. COVID-19 is a zoonotic pathogen. So here in my hand, I have data that shows that climate change is implicating the movement of zoonotic pathogens in polar bears over time. What do you do with that information? It won't be too many years into the future where you might be dealing with information that's really sensitive like this. What would you do with it? Would you take it to the media? Tell them the story? Try to influence more people to do something about polar bears and climate change? It's tempting. But one thing that the media doesn't do well is nuance. The media likes splashy headlines. They like to push forward whatever their agenda is, which can be variable. And you can lose control on a story really, really quickly. By the time something's gone viral, it's out of your control as a scientist, and you can't bring it back. The other thing is what we were looking at in this particular paper was a historical record of what's happened. And the people who are most likely to be affected by that are the people who live locally. So what we decided to do was not pursue a big press release and make a big splash about this. We went to the local communities and the local health agencies, more importantly. We provided them with common language summaries in both English and local dialects. And when we could, because it was COVID-19, we'd go and meet with the communities to talk them through what they could do to help protect them from exposure to pathogens. With that, I would like, again, to thank you for your attention and acknowledge all of the important institutions and people that were behind this work. And if I have time, I'd love to take some questions. Thank you so much.
And I've got a microphone. If anyone has questions. Way over there. I think this is on.
What did the local health agencies do with the information that you gave them?
Yeah, good question. So in the case of the two agencies that we worked with, one in Nunavut and one Nunavik, those are on each side of the bay, where polar bears are harvested by the Inuit. They've created sampling programs specifically to go out when the harvest is happening and sample from those bears to see what their levels of infection are, as well as remind the hunters of proper handling protocols, which is you're meant to take the meat and you're meant to freeze it for 3 days at least -20, and then you're meant to cook the meat after that. That's the proper way of inactivating most of the pathogens that they've exposed to. So that's what they've done, and it's been really, really successful. They do that for both polar bear hunts and walrus hunts, because walrus is another potential exposure for trichinella in particular. And so that's how they use that information, and it's been tremendously successful.
I have a follow-up on that, which is, have you detected any increases in any of these diseases in people who consume the meat?
It's a good question. What we've been tracking is the prevalence of outbreaks rather than people obviously I'd have to get their blood on a regular basis to know whether their antibodies are changing. So that's been our key metric. And what we've done is looked over the past sort of decade timeframes. And so if you go into the area where we worked in the previous decade, between 2010 and 2020, there were 3 outbreaks in the communities. 2 were related to tularemia, and the other one was brucellosis, which I didn't talk about here because we didn't actually scan for that in the bears. And so far in this decade, we've had none. And my hope is that because the information that we've been providing through this program has gone to the health agencies, and the health agencies are much more active, that's helped to reduce. But that's it's a bit crude, but that's our basic way of keeping track right now.
Thank you for the presentation. Is there a reason why the seroprevalence of virus was less affected by the climate change compared to any other types?
Yeah, that's true. So in the paper, I didn't talk about it too much here, but so are you talking about this California serogroup viruses that were lower? So a couple things. So this California serogroup viruses, one of the reasons that they're more reduced in relative to exposure is that they're mosquito only, mosquito-borne viruses. Whereas the other ones, they have different vectors. So that's one of the reasons we think, because mosquitoes are primarily transmitting it before the bears are mostly on land. Bears are starting to enter into that window where they would be exposed to it. The other thing about the California serogroup is it's actually 12 different viruses lumped together that all have the same pathway, the same reservoir, and the same sort of mode of infection, if you will. And the reason that the disease experts put them together is any individual virus in that group would actually probably fall below detection. So you're kind of pooling them to get the most out of a serum sample. And you're doing that because you're actually more interested in the mode of transport and if that's changing rather than the individual viruses within that sample. To follow up on one other thing though, so a couple we didn't talk about here, canine parvovirus and canine distempervirus. We also looked at those, and we didn't see any changes over time, and their prevalence was pretty low and just sort of continual, which suggests like a background environmental transmission that's occurring that's sort of in the system, but nothing's really changing yet. And viruses are interesting because they can hang out for a good amount of time before they find the right conditions to then erupt. That's one thing to think about when you think about pathogens. All of these life forms, bacteria, parasites, viruses, they're very opportunistic. They're just looking for that right window to erupt, right? Yeah. Thank you.
If I can get the mic to you. Yeah, you try and shout.
So the GPS trackers that I talked about? Yeah, so we deploy trackers every year. We use a mixture of the ear tags that you saw and collars. The reason I highlighted the ear tags is collars we only use on adult females because their necks are smaller than their heads. If you put a collar on an adult male, he can just push it off because his neck is actually thicker than his head. And we also don't want to put them on growing bears. So that's where we use the ear tags. So in total, we put out about 25 of those ear tags on bears each year. They last from 3 to 6 months, and then they drop off. And then we do 10 adult females, and those collars are programmed to automatically release after 1 year. And so we're continually sort of out there putting out new transmitters all the time. Yeah.
How did you end up on your career pathway?
Doing polar bears. So I'm Canadian, as you know, was introduced. When I was an undergrad, the big topic at that time was an idea around trophic cascades, at least when I did my undergrad, which is where you can take a species that has a disproportionate effect on the ecosystem, either reduce them or increase their numbers, and you have these cascading effects that happen through the ecosystem. So I was really interested in apex predators for that fact that, you know, if you change their abundance, you have these cascading changes. That's what drew me to the world of large carnivores at first. And then, well, it was quite simple. I wanted to work on the largest carnivore on the planet, and 2/3 of them happened to live in Canada. So that's how I got into it. It was not an easy, straightforward path, though. When I went to go to grad school, I had no official letter of offer. I moved to Edmonton in Alberta, University of Alberta, and I actually showed up at the prof's door and said, I'm taking courses. I'd really like to be your grad student. And that worked. But there was no official letter of offer on the table. So I kind of went the, maybe not the traditional route in order to have that opportunity. But what is really funny is you never know in life how things work out. When I finally defended my PhD, my committee had my prof, and then his advisor was part of my committee too. And we were sitting down for beers after I'd finished, and he laughed. He said, you know what? Andy, who was my supervisor, who was his student, he did the same thing to me. He showed up one day and said, I wanted to work on polar bears. And I'm like, oh, I got to take this guy. And so you never know in life how things kind of have this circularity. So that's kind of how I ended up in it. And yeah, I don't want to quit anytime soon. It's a lot of fun.
Hi, I have one last question. Does the trackers affect the polar bear's behavior, like in their natural habitat?
Does which part?
The trackers.
Oh, the trackers, does it affect their behavior? No, we haven't seen any effect, although it would be very difficult to try to commission a study because then you would have to track the other bears somehow, right? We do know that the biggest thing that we've asked is, does going out in a helicopter and chemically immobilizing a bear affect anything about their life, right? Because it's kind of like an unscheduled doctor's visit, and the bears don't, it's not the nicest thing in the world, right? But we've looked at really long-term data sets of how many times bears have been handled, what their reproductive output has been, how long they've lived, and it doesn't make any difference. So it's an uncomfortable day when we're there, and then we're gone, and they're on their way. And so that's the best information that we have to say that, no, the handling protocols that we use are okay and not affecting the bears, and neither are the GPS collars, because those bears are still reproducing. We've, like I said, put a lot of collars out on adult females, and we haven't seen any difference in reproductive output for those bears. Yeah. Thank you so much.